Needs to be loud for me. So today we're going to start with a demonstration, and in the break, we'll do a quick break. Hopefully, we'll do. You can come down and play with the microscope. That's cool. Okay, so today is going to be about fluorescence microscopy and super resolution techniques. But we have to start with the basics. So this is a very, very tiny portable fluorescence microscope. And we're going to turn on the light. Wow. This is also off. Okay, I don't know if I can turn this on off. Sorry about that. Oh, yeah, then both are muted. That's okay. I don't know how to do that. This one also muted? Yes. Okay, it will work. Don't worry. So it's more or less okay. So what you can see here, it's a slice of a plant, and you see the cursor? Yes. And you see these round things. Those are cell walls, and these things here are, well, no, these are actually plant structures, and then these round things are tiny little cells. We can magnify. Okay, this is digital magnification. There's a focus. Sorry, it's not ideal. So if I turn the focus, you see that I go through the sample. So if the focus is wrong, you see nothing. Then now it's coming in focus. Okay. And if I go too much the other way, we're also going out of focus. So a microscope has what's called a focal plane. So there's only a very thin slice in the distance with the lens where the thing is sharp. And so this is transmitted light, right? So just white light is going through into the detector. And then I can use fluorescence to see if I have any fluorescence signal. So this is green fluorescence. Not very sharp. There we go. Okay, so we zoom out. So you actually see that those smaller round things, you don't see them. Actually, plants, a lot of materials have what's called autofluorescence. So it means that there's lots of different molecules and all of them kind of very weakly fluoresce, so you can see them at different wavelengths. Okay, so this is green. I can switch to red. Okay, everything's red. And we also can go to blue. Everything goes blue. So this is a little bit boring because in autofluorescence, you see the same color everywhere. But actually this sample, if you put it under a microscope that has a spectral detector, so that can see real colors, it actually has different emissions. But in the break, we'll play a little bit. I brought the same solutions, you know, fluorescein, rhodamine. We can put some drops on a glass slide and then see what it looks like. Okay, this looks okay. So super resolution techniques, the topic of today. And of course, we have to start with some history. You all know Galileo Galilei, that made one of the first, well, not only microscopes, but you know, things that magnify. And later, this person is quite famous. This is Antony van Leeuwenhoek, and he actually was the first one to use microscopes to look at biological matter. Okay, so to remind you, we already talked about this in the first class, but some other concepts. So if we talk about refraction of light, we look at a change in the angle of the light when it enters a new medium, right? So we're going to talk later about numerical aperture, which depends on the medium. And this refraction is actually different for different wavelengths. So you all know this. So if you put light through a prism, you will get different colors that come out. And we can actually use that in microscopy. And microscopes are, of course, full of lenses, and they can be convex, they can be concave. There are all kind of more advanced lenses, but lenses basically focus light onto a single point, depending on the focal distance f. Okay, so this is nothing new. So we talked about this last class. This is again the Jablonski diagram, right? So we start in the ground state. We have all these vibrational levels. We excite to the first excited state or higher. We get this vibrational relaxation to the lowest vibrational level of the excited state. And then we get emission from this level only, which is Kasha's rule. And as we talked about this internuclear distance or nuclear separation, we can get, again, this Frank codon principle to know from which vibrational level you go. So here you go from 0 till 0, 0, or 0, 1, 0, 2. Again, relaxation and then fluorescence. And as we've seen, this Frank codon principle also is the cause of having this mirror symmetry between the excited spectrum and the emitted spectrum. Okay, so we have certain light that we excite, and the sample emits certain wavelengths. So another concept that probably you saw in high school is interference. So if we have light, here shown as a wave, and it hits two slits, two very tiny holes, rectangular holes, then each of those holes will act as a point source. So this will act like a mini light bulb. And if this is coherent light, then you will get constructive and destructive interference. And so if you look what comes out in an experiment, you see there are bright lines and dark lines. Okay, so this is interference, which we're going to talk about later. So in the human eye, of course, it's very useful. You have actually an adaptive lens. So I have some kind of object here, eye, and I look at the focal point of my lens. So that's this black dot here. And you can do these projections, right? You follow the light here, it goes to the focal point, and this one goes to the center, which goes straight, right? You know this. So this is the image which will be projected on your retina, which is smaller and it's upside down, okay? But your brain, of course, flips this. So if you have the object closer, you can actually change the focal distance of your lens. So you see that this black point moved a little bit to the left. And so you can still focus the object on your retina, even though now it's a little bit larger. Okay, so lenses and moving lenses are quite useful. So when I was focusing the microscope, I was moving the objective lens of the microscope to make sure that the focused image hits the detector. Okay, so why are we as humanity so obsessed with resolution? Okay, because interesting things happen at different scales. So at the human scale, we are roughly 1 or 2 meter size. If we go smaller to ants, they are roughly on the millimeter scale. If we go to human hairs, so the diameter of a hair is about 100 micrometer. This is just transmitted light. Even smaller, we go to human cells. They are roughly 10, these are actually 2 cells, 10 micrometers, 10 to 20 micrometers in diameter. We go even smaller. If we go to bacteria, they are about 1 micrometer in diameter. So 10 times smaller than human cells. Even smaller are big friends of the last 2 years. This is actually not coronavirus, but this is another virus. They are on the length scale of tens of 200 nanometers. Okay, so much smaller than cells. And if we go even smaller, this is a single protein. This is an antibody which has these 3 little domains, 10 nanometer size. Okay, we're going to keep going. This is a single organic molecule. So these are 5 aromatic rings. So it's in the coronine family. And this is not using optical microscopy, but this is scanning tunneling microscopy, which is we're not going to talk about, but you're in the 1 nanometer regime. So there's this huge range of sizes where interesting things happen. And the human eye basically stops at 100 micrometers. So we can see a hair, but we cannot see much more than that. So if we go to, for example, magnetic resonance imaging or PET, this is used extensively in the hospital. This is very useful, but also not very high resolution, as we'll see. And then there's light microscopy, right, which goes from, let's say, 1 centimeter to about 200 nanometers. And so there's a fundamental limit why, with a normal microscope, you cannot go smaller than about 200 nanometers. And we're going to talk about that. And we have super resolution microscopy, right? This is what we're going to talk about today, which is a kind of interesting history of how people pushed past this diffraction barrier. And of course, smaller, you can go to electron microscopy, which is very interesting, but we're also not going to talk about today. Okay. So in the last decades, there has been quite some recognition for high resolution techniques. So we're going to talk extensively about mainly the work of Stefan Hell. So Betsy, Hell, and Murner, they got the Nobel Prize in 2014 for super resolved fluorescence microscopy. And like I said, we're not going to talk about, but 2017, there was a Nobel Prize for cryo-electron microscopy. And just to give you an idea, in cryo-electron microscopy, they can do tomography, which is 3-dimensional imaging. And you can now reconstruct a protein structure from images, which I find is absolutely amazing. And it's really a revolution in biology. But not going to talk about it. So today we're going to talk about fluorescence microscopy, but just to remind you of different types of microscopy that are around. So the one I showed you just in the demo was first transmitted light, just white light that goes through. But if you think about our second class, how do you create contrast in a microscope? I just think about it. I'm going to, if I give you a very thin slice of a plant, it's maybe 10 or 100 micrometer thick. If you just put transmitted light, it might be difficult to see any contrast, right? Because you have Lambert-Beer. And if the absorption coefficient is quite similar and the length is very similar, it's going to give you similar absorption. So there are actually techniques to increase the contrast in transmitted light. And one is called phase contrast. So this means that the phase of the light also changes when you go through a sample. And actually, that's more sensitive than absorption. So that's called phase contrast imaging. And there's differential interference contrast, DIC, which makes use of 2 beams, and you send them both through the sample, and you get constructive and destructive interference, and that actually produces more contrast. So not going to talk about that. And today we're going to talk about fluorescence. And like I mentioned, you have atomic force microscopy, scanning tunneling microscopy, electron microscopy, which are very powerful, but beyond the scope of today. So why is fluorescence so cool to do microscopy? The main reason is that you can have multiple labels, right? So we can have multiple colors. We can have blue dyes, green dyes, red dyes. So similar to the experiments I showed you, so we're not going to use Schweppes, but this would be DAPI. This could be fluorescein. This could be rhodamine, right? So you can have different orthogonal labels with different colors that you can distinguish, which is quite unique for fluorescence microscopy. And this allows you to label specific parts. So if we look at biology, you can have specific dyes on specific antibodies or proteins or anything like that to stain specific parts of the cell. So here we have the the cell nucleus in blue. We have here this green spot, which is called the centrosome. This is where microtubules grow outwards, staying in green. And that actually gives you a very exquisite image that not only has resolution, but also you can distinguish different species. So let's look at some cool movies. So here you're going to look at cell division, right? So you have this mitotic spindle, which is pulling these red things. Those are your chromosomes, pulling it apart, splitting them, right? And pulling it into two new cells. So this is now, this is cell division, right? Here something is going wrong, I think, because there are three splitting. But okay, so this is incredibly powerful. A lot of biology has really been developed on the use of fluorescence microscopy and development of specific dyes, specific fluorophores that you can use for these kind of imaging. Okay, so let's look a little bit closer at the basic fluorescence microscope. So as you can see here on the table, it's what's called an inverted microscope. Inverted means the objective is on the bottom. So you're imaging usually from the bottom. Imagine in biology, you often have cells in a little Petri dish, which has liquid. So you want to image from the bottom, usually. There are new generations where you can flip the microscope upside down, so you can do inverted imaging, or you can do upright imaging. Okay, so you have the eyepieces, so you can actually look at your sample, and then usually you have a camera on the side where you can take some pictures. And so if we look at the light path, we have here, for example, a white light source, and we have a filter. So this is the excitation filter, which allows me to select one wavelength, well, not a range of wavelengths. It's often a bandpass filter, which means you cut all the wavelengths below and above, and you only save the part you want. In this case, we have white light, and we filter everything except for blue light. And this blue light travels here. It goes on this dichroic mirror, which is a very special mirror that reflects one wavelength and it transmits another wavelength. So here, blue light is reflected. It goes into the objective, that's the lens, and then it's focused on the sample, right? So this is the focal spot, and then the sample will emit light, green in this case, so it emits green light. The green light goes back into the same objective, so it goes back down, and then it goes here, and now it goes on the dichroic mirror, but in the range where it's transmitted. So the green light goes straight. Here, there's another emission filter. Why? Because there's always some scattered light, right? So if you emit blue light, or sorry, excite with blue light, and your sample has some scattering, which it always will have, there will be also some blue light that goes back into your objective. So you want to reject that light, which happens on the dichroic, but to be sure you have an emission filter to only pass the green light, and then it goes, well, either into your eyes or into a camera. If you have any questions, please interrupt. So one step up from the fluorescence microscope is called a confocal microscope. So in a confocal microscope, you have something new, which is a pinhole. And so what does this pinhole do? So here we have our sample. So now everything is twisted 90 degrees, right? So we have our sample and we have our focal plane. Okay. So in a normal microscope, you would have these blue lines and not a pinhole. So you would see light that is coming from the focal plane, but also from planes that are higher and lower, right? So if I have a thick sample, I will see light from different layers in my sample. So in confocal microscopy, you use a pinhole, and that pinhole only allows you to capture most of the light from a very, very narrow slice. Okay. So I have this information now, which allows me to get less background signal. So I only get a very thin layer, so more contrast. And I can actually move my lenses to get information from each of these layers. So I can reconstruct a three-dimensional image by scanning through my sample many times, taking at each focal plane an image, and then reconstructing it. So for example, this is a slice from a zebrafish. It's colored in red and green, and you can reconstruct and get this three-dimensional image of inside this zebrafish, which is pretty cool, right? 3D information, of course, tells you a lot more than a flattened image. Okay, that's confocal microscopy. So let's talk about that in a bit more detail. So in the introduction, I told you there can be interference, right? So if we have a pinhole, so now we have not a slit, which is a rectangle, but we have a round hole, which is the pinhole, then we will get a similar kind of interference pattern. But now this thing is called an Airy disk after Mr. Airy. And so you get, again, destructive and constructive interference, and you get a pattern that has an illumination pattern, something like this. So in the center, it's the brightest, and then you have these kind of waves around where you have first destructive and then constructive interference, which are alternating. So the shape of this intensity profile is called the point spread function, the PFS. Okay, so if we cut through this point spread function, so I'm going to take this plane, I'm going to cut here, then I will see this blue profile. So I have the largest intensity in the middle, and then we have these smaller bands that go around. So now imagine you have two molecules that are dyes, and they are emitting light. I want to know like the shortest distance they can be where I can still separate them, right? That's what determines your resolution, and that's called the diffraction limit. So if I have two dyes, and they're usually the same color, right? But to make it easy here, they're blue and red, and they're far apart, I can easily distinguish them, right? Because if I measure the intensity profile, I have 100%, 0, 100%, 0, or close to 0. But if they come closer, I can no longer distinguish them, right? So what's limiting? Why is my image sometimes blurred? Because I cannot distinguish two molecules that are very close. And so two people have postulated, or actually, for many hundreds of years, this was accepted as a total truth, that this distance where you can still resolve two dyes depends on lambda, which is the wavelength of the light, and NA is the numerical aperture. And so this is almost the same if you compare these, right? This is 1/2, so half lambda, and this is 0.6 lambda. So this was said by Lord Rayleigh, and this is said by Ernst Abbe. And so it seems that there is no way, based on this physical laws of light, that you can get a resolution which is, let's assume NA is 1, numerical aperture of air is 1. So lambda/2, if we use 400 nanometer light, the maximum resolution we can ever get is 200 nanometers, which is nice, but we'll see that we can go smaller. And some people are very serious with their work. So on the gravestone of Ernst Abbe, he engraved, people engraved his equation, right? So diffraction limit is basically lambda/2 under some assumptions. Okay, so because these were very well-established people, there were very few attempts to actually get better resolution until the last decades. So this has been very exciting time for super resolution microscopy. And we're going to talk about all of these little fancy acronyms. So STORM, PALM, PAINT are specific super resolution microscopy techniques, but they fall basically within 3 different categories. The first one is called structured illumination microscopy, or SIM. Then there's stimulated emission depletion microscopy, STED, or single molecule localization microscopy, SMLM. Okay, so we'll dive into those different techniques. Okay, so structured illumination is kind of the hardest one to explain, so I'll try to go slow. Basically, we have to understand first that we can look at information in the spatial domain, which is what you normally do when you take a photo, but we can do a Fourier transformation to the frequency domain. Have you learned things about Fourier transformations? Yes, people are like, yes. Doesn't sound very enthusiastic, but yes. Okay, so Fourier transformation is very powerful. We'll see it later in infrared spectroscopy as well. But we're going to transform information from the spatial domain to the frequency domain. So in these images, what actually are you seeing? So in the very center of the image, you have to think of it as a radial map, basically. So the center has low frequencies, and the more you go outwards, you go to higher frequencies. So I can split this spatial domain into a sine wave, right? So I have low, high, low, high. And I map this on the frequency domain, so I'll have some low frequency and some higher frequencies going outwards. And the intensity tells you something about the amplitude of the waves. Okay, so if I look at this image, I have some orientation and I have a different spacing, so I get 3 spots in my Fourier transform. And if I superpose this image and this one, so you see some lighter lines like this and some fainter lines like this, I can actually see that it's a superposition of these images. Okay, so Fourier transform, you transform from the spatial domain to the frequency domain. It's fully reversible, so I can do inverse Fourier transformation and get my information back. Okay, so now you have to think about any optical system. And we're not going to talk about these optical transfer functions and things like that, but just think about a Fourier transformed image and your optical system. Because of the wavelength, because of the numerical aperture, you can only cover a certain area in your Fourier space, right? So I have my lowest frequencies here, and I go outwards to higher frequencies. And at some point, I have the diffraction limit, right? So the diffraction limit, this is 1 over the equation we just saw. So the radius of this disk depends on the diffraction limit, which is 2 times numerical aperture over the lambda of the emitted light. Okay, so let's do it in a different way. So you have this lovely gentleman here. We do a Fourier transformation. Okay, so the guy, this is the same thing. Of course, if you share this on social media, people are like, what is this? But this is the Fourier transform of this image. And we are going to take the image with a certain camera, right? This camera will have lenses, it will have some kind of wavelength to detect my image, and therefore there will be a diffraction limit or a limit on the optics of that system. So I'm going to lose my higher frequency information. So that will be black, right? So I just take this image, I crop a circle and delete all this information here. And now I do the inverse Fourier transformation. What do we see? The image is more blurry, right? I lost resolution, Actually, if I only Fourier transform the remaining information, we can still see some kind of features that were lost. Okay. So talking about moire patterns. Who knows what moire patterns are? Okay, it's this thing on TV, right? If you go on TV and you wear a shirt with squares, or you have a very ugly tie with squares, it kind of makes this weird things, right? Those are moire patterns. I'm assuming it's a French person, but okay. So we have a projector here, right? So these are sheets which have periodic lines. And if you put two of those line patterns on top of each other, there's no actual lenses you can. So, but you see the patterns you make, so the moire patterns, they are much larger than the features of the individual sheets. Right? So, okay, so this person is just sliding these sheets, and that's creating these moire patterns. Okay, I just really like this movie. Okay, so moire patterns, they're pretty cool. So how can we make use of this phenomenon, of these moire patterns in microscopy? So imagine we have a sample that looks like this, and we shine light, which is not uniform, but which also has a pattern. What we're going to end up with is moire patterns that have features that are much larger than the small structures that we're trying to see here. And so these moire patterns, there's a lot of mathematics behind this, which we're not going to talk about. But depending on the angle of this grating, well, these high-low intensities, we can get different moire patterns. And so if you illuminate your sample with this kind of grating, record the moire patterns, you can reconstruct higher resolution information than you had originally in your microscope. Okay, this is a bit technical, sorry about that. So in this setup, you have a laser. It goes through a lens, a pinhole, and then this grating here is a microstructured piece of optics, right? And so I will split my beam into smaller features that are then going through a lot of optics and go on my sample. So then this is recorded, and then you image at many different spacings and orientations of this grating. Okay, so what does that represent in Fourier space, which is again quite technical. But remember, so if we are in Fourier space, we have the low frequencies and increasing frequencies outside. And we have the diffraction limit. So what effectively happens when you use structured illumination is you can actually shift the position of this disk. Okay, so I take my resolution disk and I move it around to the boundaries of my Fourier space. And by changing the angle, I can walk with this disk around the circumference of this circle. And that actually gives you a larger area in the Fourier space that is now accessible. So effectively, you're increasing the information in the Fourier space, which means you're going to get more resolution. It's about 2 times higher, which is already quite amazing. Okay, so that looks like this. So if you would look through the microscope, you would see some kind of cells, and you see these squiggly lines on top, which creates these moire patterns. Okay, you won't actually be able to see or extract any information from this by eye, but after mathematical processing, you can now reconstruct this information and get higher resolution data. So what does it look like? You know, it's microscopy, so we have images. So this is on a normal fluorescence microscope, and this is using structured illumination. So that's quite a big improvement in resolution. Okay, here another example. So this is normal confocal microscopy. This is SIM, right? Again, much higher resolution, much sharper features, and some features where you would not be able to distinguish them. Then with SIM, you can actually really clearly see a difference. Okay, so the benefit of SIM, of structured illumination, it's, well, simple to execute. Maybe conceptually it's not that simple, but it's simple to execute. You don't need any special dyes, right? Just any fluorescent molecule will work. It's quite fast because the illumination can be switched very rapidly, and you don't need much light on your sample, which are all great for biology. In biological samples, you always have to think about the total amount of light you're putting on your sample, because if you put too much light on your sample, what happens? Well, it explodes when you put really a lot of light, but if you put normal amounts of light, what do you get? You get photobleaching, right? So photobleaching means your dye does some kind of chemical reaction with something like oxygen, and it will break the fluorophore and your fluorescence will decrease, which can be useful. We'll come back to that later. So here we get 2 times increase in resolution, but we cannot go more than 2 times. Okay. Okay. So this is Stefan Hell, and he has his own little equation. Okay, which is if you recognize it, is basically the same as the Abbe equation. This part. This is the d, the diffraction limit. But there's this new term, which is square root of 1 plus j over js. Okay. What does that mean? And for that, we have to go back to the first class, where we talked a little bit about stimulated emission. Right? So in stimulated emission, we have first absorption, right? We can have emission back to the ground state. And that depends on this energy difference between the states, which is h nu, right? We talked about this. But what you can also do is you can use a different wavelength to stimulate, to force the emission back to the ground state, which prevents the dye to emit at the wavelength where it was fluorescing normally. Right? So I can use a second beam of light to deplete, to kill the fluorescence of my first dye. Okay. So here we have normally, for example, in confocal microscopy, we have the excitation beam, which has a certain width, right? Which is limited by your optics. So what they do instead is they have a second beam, which is called the depletion beam. And this depletion beam is like a donut. Okay, so it's light that using special optics, a special method, they can make this donut. And if you put them on top of each other, you have the excitation beam, which is surrounded by the depletion beam. Okay, so what is the result of that is that the point spread function, right, this PFS, PSF, yeah, is actually smaller than it can optically be. Okay, so in more images, so the excitation beam has this kind of Gaussian shape. The depletion beam is a donut. I put them together, and I only get fluorescence emission from a very narrow region in the center. Okay, so if we look at this in the time domain, right, this we said in the previous class, excitation, if you use a very short pulse, you will have some delay, and then you have fluorescence, which decays, right, using a first order reaction kinetics. So here we add this second stat pulse. So we excite, we shine the donut a few picoseconds later, which is very short. And then I will lower my fluorescence, but also more importantly, I will have fluorescence from a much smaller spot than initially. Okay, so we'll look at this in this movie. So you'll see first a normal confocal beam, right? So you have the objective, you have the focal point, and we'll scan these two molecules with the microscope. So normal confocal microscopy, you have the confocal volume, and we're going to move the beam and measure the intensity. So I get the first molecule, the second molecule, but because they're very close, you will only see one peak, right? Now, if we turn on the depletion beam, which is the red thing, I make a much smaller excitation beam, and now I can see my molecules individually, right? So this explains why the resolution in STED is much higher than in normal confocal microscopy. Okay, so the cool thing instead is that you can actually change the resolution by tuning the power of this depletion beam. So if I increase the power of this donut around the excitation beam, I can go from a resolution of 200 nanometers, which is the normal confocal resolution, and I can go all the way down to 50 nanometers, which is much, much sharper. But there's, of course, a caveat in all this. The caveat is back to this equation, right? So this is the resolution, wavelength of emission. This is all the same as the Abbe equation. And now we have this new term, which is the power of the excitation beam over that of the saturation of the depletion beam, right? So by increasing the power of this donut, I can lower my resolution. And so just as for information, in a normal confocal, the power is around kilowatts per square centimeter. So to go to 50 nanometer resolution, we already need 214 megawatt. So megawatt is already a lot, right? So so here, if you see this marble cutter, that's megawatt. So if you want to really cut, metal in half, you can use those intensities. Of course, it's pulsed, right? Let's go back to you only have a very short pulse, so it doesn't mean your sample will evaporate. But still, you have to be aware that you need quite special equipment and very heavy lasers to reach these kind of resolutions. Okay, I'm actually going much faster than I thought. So I think we can skip the short break, finish the class, and then for whoever wants to play more with the microscope can come and play with the microscope. Okay, so some images, right? This is normal confocal microscopy, and this is STED. So you see these red structures are much more resolved than the green structures. Okay, this is also used in supramolecular chemistry, which is my field. So I like this stuff. So here there's an example of supramolecular fibers. Hello. Okay, let's just focus for 10 more minutes. So here you have these fibers, and there's red fibers and green fibers that coexist. So in this case, this is a fluorescein type dye, and this is an Alexa 546 dye. So it's a red dye and a green dye. And if you do high resolution imaging, like super resolution imaging, we can actually distinguish fibers that are red and are green. So this means that the supramolecular fibers, they are self-sorting. They form their own fibers where all the red ones go in one fiber and all the green ones go in another fiber. Okay, so the advantage of STED is you have good resolution. It's quite simple, right? It's fast. You can use it on live cells. Still, it's not, now it's commercially available. So if you have enough money, you can buy it. It's very expensive. You need very high powers. But also and we didn't talk about this, you need special dyes that can actually be depleted. So not all fluorescent molecules, when you shine a depletion beam, you can actually kill the fluorescence. Okay, so it's limited to special dyes. Okay, so this is Stefan Hell in the middle. This is John Pendry, and this is Thomas Ebbison, who used to teach this class. Now you're stuck with me. Okay, I'm also Thomas, but not Ebbison. So Thomas Ebbison got, together with Stefan Hell and John Pendry, the Kavli Prize. So the Kavli Prize is like the Nobel Prize for nanotechnology. And he hasn't worked on super resolution techniques, but on extraordinary light propagation through small holes. Should definitely check out the work of Professor Ebbison. Okay, last technique, which is called single molecule localization microscopy. So what is this? It's very smart because you don't need any special optics in your microscope. It uses a normal fluorescence or confocal microscope. And so let's imagine you have this little worm here, which is fluorescent. And if you take what's called a wide field image, which is what we were doing here in the demo, all the dyes in your sample will emit at the same time. So I will see a worm, but it will be blurry because all the dyes are overlapping. So now what's happening in localization microscopy is that you try to get a situation where the dyes are not emitting at the same time. So how can you do that? That sounds kind of difficult. And it is. So we'll see there are different approaches to get to that point. So basically, you will take a lot of images, and in each image, there will be different dyes that are illuminated, right? So you can, if you have only one dye emitting and no other dyes close, you don't care about this diffraction limit, because I can still localize each of these points by fitting a Gaussian function on each of these spots. Okay, we'll see what that looks like, but basically I can determine the location of a dye much more accurately than I can distinguish two dyes from each other. So if I then sum all these locations of all these dyes together, I can actually get a higher resolution image than when I compare it to wide field. Okay, so let's look what that looks like in the microscope. You will just see crazy blinking of stuff, right? And this is what the reconstruction looks like. So after some time, you will actually be able to determine, let's go back and look at this movie again. It was a bit fast. So if I look at each blinking event, I can draw a Gaussian function here. So I can determine the center of that spot very accurately. So that's the whole trick of this technique, right? Oops, play, no. Sorry, go back. Yes, so all the dyes are blinking. I determine the location. I put a little dot at the center of the location, and therefore I get an image which is much higher resolution than my actual microscope gives me. Okay, this is a little bit technical. So this is the precision. It's similar to the resolution, which depends on the standard deviation of the point spread function. That means how broad is this Gaussian distribution that the light is emitting, right? So if that's very broad, your resolution is worse. That divided by the number of photons you collect. Okay, so more light, you get better resolution. Of course, the pixel size of your detector, so a detector also has pixels. So if you have smaller pixel size, better localization, of course. And this last part, which is kind of annoying, this is basically the noise of your detector. So even good detectors, if they receive no light, no photons, they will still give some signal. So you get some electronic noise, which lowers your resolution. Okay, so that's, let's say, an instrumental, technical limitation. What's also you have to think about is that fluorophores can be small molecules, but can also be fluorescent proteins, right? GFP, green fluorescent protein, also got a Nobel Prize for that one, is a protein that you can express. Well, not normally. I actually, one time, I accidentally transfected my finger with GFP expression. So I had a fluorescent finger for a couple of days. Don't try that at home. Okay, but GFP, it's a very intensively used protein in biology. And often biologists, they really like to use antibodies because antibodies are very specific. They can bind to very to targets with very high affinity, but they're also very large. So if you do, let's say, an antibody recognition, this is what's used in an ELISA test, for example. This whole thing is already 20 nanometers. And this is floppy, right? Molecules are floppy. So if you attach this and you want to image it, you already have, the maximum resolution you will get is never smaller than 20 nanometers. So you have to think about the size of your fluorophore. Okay, this doesn't really matter, but of course, in a microscope, if you image at nanometer resolution, any small change in temperature will move your sample, right? So you don't maybe think about that, but if you have a 5-degree change in your room, all the materials will kind of expand and contract, and this will lead to small movements, right? So you have to correct for this small drift in your system. Okay, so I told you you need dyes that blink. So I'm going to tell you 3 different methods for getting blinking of dyes. And the first one is called STORM, which stands for Stochastic Optical Reconstruction Microscopy. And in STORM, you have a dye which has 2 states. It's on, it's fluorescent, or it's off, it's dark. And I can use reduction and oxidation chemistry to switch the dye on and off. So this is reversible photo switching. So let's look at an example. I have a dye here, which is fluorescent. And I have a thiol, this R group with a deprotonated thiol, and that reacts with this double bond here, and that breaks the conjugation, and my dye is dead, right? So I don't have any fluorescence emission here. But this is reversible, so if I shine light, I can actually reverse this reaction, and I go back to my fluorescent molecule. So I get a dye that's blinking in time, which I need in my microscope. Second method to do blinking is PALM, which is photoactivation localization microscopy. So you first have a dye which is in the off state, and you use typically UV light to turn the dye on. And then when the dye is activated, you can image it for some time, and you keep shining light, you will photo bleach the dye and it will go off. So let's look what that looks like. So I have my initial view. I shine with my activation beam, which is UV light. A few dyes will go on, these 3. Then I read my fluorescence. I see these 3 spots, and I can localize them very accurately. And then I keep shining light, and I kill them. So photo bleaching with long illumination, it will turn the emission, or basically will destroy your fluorophore, and therefore it will go off. Then you repeat this, so you shine again your activation beam. Now you activate different dyes. These were the ones that were killed. And now I can localize these ones. And you repeat this over and over, which allows you to localize each of these molecules individually and therefore get high resolution. Okay, so this is an example. For example, this is fluorescein, but it's photocaged. This is a very famous leaving group. So if you shine UV light, you will deprotect these moieties here, and you will get free fluorescein, which is green fluorescent. right? So if I have this square, I shine my UV light, I turn on my fluorescence. So this is a very nice approach to switch on my fluorescence and then kill it with photobleaching. Okay, paint, last one. So in paint, the dye is always on, but the emission depends on whether the dye is bound to the target or not. So for example, if I have a flow cell and I have my dye in solution and I have this star here, and the dye can bind to the star, it's fluorescent when it's bound. And then when it unbinds, you don't see it anymore, right? So I will still get fluctuating intensity of fluorescence on this structure, and that gives me high resolution. So this is an example. Here I have some fibers, and I use this double antibody, which biologists like, and I have DNA, which is complementary with this strand, which is on the antibody. And this dye is only fluorescent when it's in the bound state. So when it binds, the fluorescence turns on, I can image it, it unbinds, it flows away, and the fluorescence is off. So therefore, this would be diffraction limited image, using normal fluorescence or confocal microscopy. And this is my paint image where I get super resolution because of binding and unbinding of the fluorescent dye. Okay, so this is very small to read on the screen, but on the slides it will be nice. It's an overview of the different techniques I talked about: localization microscopy, stimulated depletion, and structured illumination. And so you can see that the resolution limits are getting better and better. So for example, with Palm, Storm, I can go to 10 nanometers. It's very slow, right? So if you have, because you need to take 1,000 images and then localize them. So the imaging time is a bit long, but the resolution can be really, really great in this case. So depending on your application, depending on the resolution you need, you can choose one of these techniques. Any questions? Okay. If not, thank you. And for those who want to do the demo here, you can come down, and the rest I see you in the next class.